Hello everyone, and welcome to Meiosis Made Simple. And this video here, if you watch it, will definitely make the week of meiosis a whole lot easier. So in meiosis, which is a little bit different from mitosis, you are going to start with a cell, which is going to be a diploid cell, but instead of making two identical copies, you are going to make four genetically different copies, and each of those four will have half of the chromosomes that you started with. So it's important to understand that only special cells can do this, and they are known as either spermatogonium in men, which will form sperm, or oogonium in females, which will form eggs. So our diploid number is 46 chromosomes, but for this case, we're going to start with a simple example and say the diploid number is 4. And remember, that is the total number of chromosomes to start. So in this case, we're going to call this one Mom 1. We're going to call this one Dad 1. We're going to call this one Dad 2, and this one Mom 2. So a couple of things we have to get straight. These two, whether it's D1, M1, or M2, D2, whatever, these are homologous. So homologous means that they are the same chromosome number. They contain the same genes, but they are not identical. So you get one from your mom, one from your dad. A second thing I'm going to introduce is something called a picogram, which is in essence a weight or a mass for something. And it's a very small mass. And it'll work well here for us. So there's 30 total picograms of mass here, or picograms of DNA. We're going to keep it simple, and we're going to say the large chromosomes that I draw, that one has a value of 10 picograms. That one has a value of 10 picograms. These guys are smaller, 5 picograms and 5 picograms. So your total mass is 30 picograms. This is your normal cell doing normal cell things. Next, of course, is the S phase. Now, the S phase is going to happen in both mitosis and meiosis, and that is where you will replicate your DNA. In essence, you are going to double the amount. Now, that is important to understand. So if you had 30 picograms, now you're going to have 60 picograms of total DNA. Let's see what happens. Prophase 1, you're going to have your homologous chromosomes that have now been replicated, because once we get to prophase of anything, whether it's mitosis or meiosis, we need to know that the interphase stage or the replication phase has already happened. In meiosis, that replication will happen only once in interphase 1, in essence. So here you're going to see homologous chromosomes interacting and exchanging genetic information. Now watch how it goes. You know you're in prophase for a couple of reasons. 1, the chromosomes have condensed so you can see them. 2, the chromosomes have been replicated and they resemble the letter X. When I draw them, a lot of times I'll draw them like this with a 1, and then I'll have the second one come down like that, and then I'll have them held together at a centromere. Here I use color so you can see. These are technically called sister chromatids because at this point they are identical. So just to kind of keep an eye on what's going on here. These would be considered sisters, but the blue and the red would be considered homologous, and that's important to understand. So let's see what we got. We are technically still diploid. We still only have 4 chromosomes. But since they've been replicated, they are now called double-stranded. Do not say you have 8 chromosomes, because you do not. You have 4 chromosomes that have been replicated. Because remember, this is Dad's 1, this is Mom's 1, this is Dad's 2, this is Mom's 2. You did not add any more chromosomes. You simply doubled the amount of DNA. So now your total mass is 60 picograms. Each homologous pair, since it contains 4 total chromatids, are going to be known as tetrads, because tetra or tetrad is going to resemble something having to do with the number 4. So right now, you know you're in prophase. Right now, everybody has been replicated from interphase, and this looks very much like mitosis. But now things are going to get a little different. there's going to be a process known as synapsis. And what's going to happen there are these homologous chromosomes are going to interact with each other. And the spot where they interact, let's say right there and right here, is going to be known as a chiasma or chiasmata, as you may see it. Once again, I still have my diploid number of chromosomes. I still have 4. They are still double-stranded. Nothing changed. I still have 60 picograms. I am still in prophase 1. Prophase 1 is the most important step of meiosis having to do with genetic diversity. Now, when the homologous chromosomes pull apart, they have in essence swapped some genetic information. And if you look very closely, you have a little bit of blue at the bottom of the red and a little bit of red at the bottom of the blue. And you can see here that I swapped out the green and the purple. Again, I still have 4 double-stranded chromosomes. I still have a total of 60 picograms of DNA's mass. Still, I haven't added anything. I haven't taken anything away. I've only swapped. I'm still in prophase. Prophase 1, and again, crossing over is huge when it comes to prophase 1 and genetic diversity. Now, we're still in meiosis 1, and again, meiosis has 2 phases if I didn't mention it earlier. There will be a replication of DNA followed by a division of cytoplasm, and then in meiosis 2, you will simply have a second round of cytoplasmic division. So we're still in meiosis 1. Now, what's going to happen here is the chromosomes are going to line up along the equator of the cell with the metaphase plate, but they're going to do it differently than they did in mitosis. In mitosis, they all kind of lined up kind of like on top of each other like that. So this would have been, let's say, dad's 1, and this would have been mom's 1. But look at how they're different here in meiosis. The homologous pairs are next to each other. That is a huge difference, and it's going to play as we watch these cells divide. So remember, this is your dad's 1, your mom's 1. Your homologous pairs are next to each other. Your dad's 2, your mom's 2, next to each other. You are still diploid number 4, still double-stranded, still 60 picograms. Nothing has changed. The only difference between metaphase 1 and metaphase in mitosis is now, again, the homologous pairs are next to each other. I cannot stress enough how important that is. I didn't draw these in all the other steps, but you're still going to have your centrioles, and they're going to be at the poles, just like everything else. And what's going to happen is the spindle fibers are going to come out, and they're going to grab the centromere, just like they did in mitosis. But instead of taking the sisters apart, you're going to take the whole homologous pair. So your anaphase 1, the homologous chromosomes, you'll see them are going to travel together to the poles. This, again, I cannot stress the importance of this step. So watching, once again, if I put my centrioles there, there's my spindle fibers, the whole kinetochore thing, everything's exactly the same as it was in mitosis. So now, again, the homologous pairs are separating. I have not divided anything yet. I still have my diploid number 4. They're still double-stranded. Everything's cool. I still have 60 total picograms. Nothing's changed. Now I'm going to bunch together telophase 1 and cytokinesis just to speed this party up. But watch what's going to happen after this step. At the end now, you're going to end up with 2 cells, kind of like in mitosis. Now here's where you really got to pay attention to the number and the vocabulary. Each cell is going to contain 2 double-stranded chromosomes, but now I'm going to tell you that the cell is going to be considered haploid. And once again, haploid means instead of having 2 copies of each chromosome, you're only going to have 1 copy of each chromosome. And now, and this is again pretty interesting, you're going to end up with the same amount of actual DNA mass as the original cell. So if you remember, the original cell started out with 30 picograms. Watch what happens here. There's your 2 cells, Each one is haploid, and here's why. This is your dad's 1 with a little bit of mom. This is your mom's 2 with a little bit of dad. This is your mom's 1 with a little bit of dad. This is your dad's 2 with a little bit of mom. Notice 1 of each chromosome, even though they're double-stranded. So the haploid number once again is 2, double-stranded, 30 picograms. And again, in case we forgot where that came from, this long one, even though it's blue and red, was 10. This one's 10. This one's 5. This one's 5, for a total of 30 picograms, exactly the same mass as you started. Now, obviously, the orientation is different. Everything's totally whacked out, but the amount of DNA is the same. Now, we're going to jump right to metaphase 2 because interphase 2 and prophase 2, nothing big happens there. It doesn't really matter. But now is where you got to really pay attention to what's going on. Now, in each of these cells, these kind of mixed up non-homologous chromosomes are going to have to split again. And this is where we're going to see some similarities to mitosis. And the only similarity is the order of the way things are going. And again, I'll explain what that means in a second. So once again, here's the cells that we left off after telophase 1. Each one has 30 picograms. Each one's haploid. Each one has 2 double-stranded. But now notice the chromosomes are lined up on top of each other. They're in a row, just like they were in mitosis. So here is a similarity, at least in the way that it's designed. So again, we can't forget about our centrioles because they're there. Everything's the same, the whole centriole spindle fiber thing. And now it's going to act very much like it did in mitosis, where you're going to now separate the 2 chromatids from each other. But again, the metaphase is important because it looks similar to the way it did in mitosis. So anaphase 2, what's going to happen now is you're going to separate these chromatids. They're going to move to opposite sides or opposite poles, just like in mitosis. But check out what happens now. Now you're going to see that this is going this way, and this one's going this way, and this one's going that way, and this one's going that way. They're moving away from each other in anaphase like they're supposed to. Same thing's happening here. So hopefully you can start to see that this is going to set up another cell division. But notice I did not replicate the DNA. So by the time I'm done, I'm going to end up with 4 cells. And that's the take-home message here. Telophase 2, cytokinesis 2, big deal. You're going to end up now with 4 cells. Haploid in nature, meaning 1 of each chromosome, either mom's 1 or dad's 1, mom's 2 or dad's 2, and so on. Now, of course, in yours, you're going to have 23 copies, but we're keeping it simple here. In this case, they're going to be single-stranded only, so there's our half. And because there has been some swapping of DNA somewhere, each of these cells will be genetically different from each other. So let's check it out. We got... Okay, that whole top is the same thing. The key here is that you're now going to have single-stranded chromosomes in each cell, and each cell is only going to be 15 picograms, which again is half of what you started with. So let's kind of see what happened here. We ended up with all of dad's chromosome 1 here, most of mom's chromosome 2 with a little bit of dad. This is most of dad's 1 with a little bit of mom, all of mom's 2. Over here we had mom's 1 with a little bit of dad, all of dad's 2, all of mom's 1 by itself, most of dad's 2. And the key that you have to see here, it has to be super obvious. A, you started with 4 total chromosomes, now you have only 2. You started off with a complete red, a complete blue, a complete green, and a complete purple. Now you have this mismatch of either all blue, blue and red and blue, purple, green, whatever. And notice that each of these is genetically different. Now this is going to happen each time any spermatogonium or oogonium goes through meiosis. That's why you will look similar to your brothers or sisters, or you may look totally different from your brothers or sisters. So hopefully, in conclusion, you will see that in mitosis, you went from one cell to two, and these were genetically identical to each other. And in meiosis, you went from one cell. Now, this is a special cell, by the way. It does not happen in all cells, just the special cells. You're going to have a replication first, followed by a division, just like in mitosis. I should have said that. A replication followed by a division. And you're going to end up with your two cells. Then you're going to have a division again, and that's where you're going to end up with your four cells. But because you're dividing without replicating, each of these cells will have half of the number that you started with. And because of the whole prophase 1 crossing over, they're going to all be genetically different. So hopefully this is helpful and will make the rest of this week a little bit easier for you.